I hope you enjoyed the Nomenclature Part 1 video, as well as the live online classes. And now, to kind of summarize week 1, I want to look at that very important nucleophilic attack step and to look at some examples of applying organic nomenclature. Again, whenever there are blanks to fill out, questions to answer, concepts to discuss, things to sketch, please go ahead and make those notes, which will need to be submitted by midnight on Saturday. For Nomenclature Part 2, I have the same learning objectives as in Part 1. And I want to remind you that the reason we're looking at this organic nomenclature is because they are the syllables used to build up biochemical nomenclature. And you will find this whole course a lot easier if you can really try to understand how to apply organic nomenclature. To simply memorize a whole bunch of biomolecules and proteins and pharmaceuticals as if they're just a group of syllables which you don't understand would be very painful. So let's move on. Life can be explained by a handful of simple chemical reactions. In particular, we are constructed out of simple building blocks, such as nucleotides, amino acids, and monosaccharides. In order to make something complicated out of simple building blocks has to do with how they're put together. For example, there's only 4 nucleotides, A, T, C, and G, but in a genome, there are 3 billion of them linked together. Just as the 20 different amino acids can be linked together in sequences of 100, 500, 1,000 to make all sorts of different proteins, so the 24 letters in our alphabet can be linked together to make words, sentences, paragraphs, books, text messages, fake news. There's many types of monosaccharides, but there are even more different types of ways to link them together. And my assertion is if you understand the building blocks and how they're linked together, you will understand life. Let's review a little bit from Part 1. Will you go ahead and use Greek lettering and Arabic numerals in order to designate each carbon in this structure? Please pause and do so. Well, of course, we looked for the landmarks. Oxygen is the biggest atom, nitrogen next biggest, then carbon and hydrogen. The number 1 carbon will have to be by the biggest landmark. It'll be linked to the carbon in the carboxylic acid. 1, 2, 3, 4, 5, 6. How about the Greek nomenclature? Please go ahead. I'm only going to be worried about carbonyl carbons. This is the carbonyl carbon, so it has a label. The alpha carbon will be next to it, alpha, beta, gamma, delta, epsilon. And so you'll notice that the number 2 carbon corresponds to epsilon, number 3 to beta, 4 to gamma, because the alpha carbon is adjacent to the number 1 carbon. By the way, do you recognize something about this compound? First of all, what is the general category of compound and why is this important? And second of all, what is the specific compound? Well, I'm hoping that you recognize that it has an amine and an acid, hence it is an amino acid. And the side chain is positively charged. This turns out to be lysine, and it is one of the important amino acids. Now, to be clear, I do not think you should know the structures of all 20 amino acids, but it will help a lot if you know about half of them, and we will talk about those in a future video. I'm pointing out lysine now since we are using it as an example for numbering carbons. But now I want to move to a different compound to emphasize how some familiarity with organic nomenclature will help you remember a lot of pharmaceutical nomenclature. It is very hard to memorize gibberish. It's easier to memorize words when you have some idea of their meaning. Right now, I would like to take a look at a very important pharmaceutical, acetaminophen. This is the most common cause of acute liver failure in America, even though it's one of the most widely used and trusted pharmaceuticals out there. The purpose of my looking at acetaminophen is to get you to think about the organic nomenclature that underlies all sorts of enzymes, pharmaceuticals, metabolism. You should not memorize the structure of acetaminophen. You don't need to know what organic group is connected to what. But can you correlate each syllable in the name of the drug with the structure? Please pause and do so. But I'm going to move on. Acetaminophen. We have acetyl group, amine group, hydroxyl group, phenyl group, acetaminophen. It turns out that each of these is profoundly important in all sorts of drugs. A lot of drugs have extra phenyl groups in them because they're greasy and they help cause the drug to stick to its target. The phenyl group also plays major roles in human biology. You should always recognize phenyl with any name. Of course, hydroxyl groups, that'll have a lot to do with making drugs soluble. The acetyl group is probably the most important group for you to be completely familiar with. Why? Because the addition or removal of acetyl groups to all sorts of biological molecules, to all sorts of carbohydrates and lipids, is at the heart of metabolism and human signaling. You really want to know what an acetyl group is. And in fact, it's so important that I'm going to look at another compound where it plays a major role. Some of you are probably familiar with the compound acetylcholine. Will you pause the video and tell me what it does, and also will you associate the parts of the chemical nomenclature with the parts of the structure? Please go ahead and do so. So again, hopefully you're beginning to recognize the acetyl group. Whenever there's an oxygen interrupting a carbon chain, you know that something is linked there. So the acetyl group is the group to the right, and choline is the group to the left. Acetylcholine literally means acetate linked to choline. And any time you see YL in the middle of a chemical name, throw in the interpretation linked to, and it's a lot easier to understand a lot of complicated terms. Acetylcholine is a pretty important neurotransmitter. Also, it turns out that the choline part of acetylcholine is an important nutrient, and it's attached to a lot of fatty acids. So choline plays a lot of important roles, and you'll want to know it. But even more important is the acetyl group, which is central to energy metabolism. In fact, the addition of an acetyl group to a compound or hydrolysis of an acetyl group from a compound plays profoundly important roles. Will you draw two arrows to show the movement of electrons in the nucleophilic attack of a water molecule on acetylcholine? I believe the next 5 minutes of this talk represents my fundamental philosophy and strategy toward teaching biochemistry. I focus on simple fundamental concepts which can be broadly applicable. A nucleophilic attack mechanism is something that you've absolutely studied a number of times in organic chemistry, but maybe don't remember all the details. So again, please draw the arrows. In any nucleophilic attack mechanism, you look for two things: a double bond and a lone pair of electrons. The lone pair of electrons may not be shown, so you look for an oxygen, nitrogen, or sulfur. The double bond is going to be attacked by the pair of electrons. So we can connect a lone pair of electrons on a water molecule to the carbonyl carbon. And there's always going to be two arrows. The second arrow will go from the double bond up onto the oxygen. After the oxyanion forms, the electrons will resonate back down and kick off the leaving group, in this case choline, which will pick up a proton. And let me be clear about that proton. I don't care about it. You know, in organic, they say, hey, is this donated from a group on the enzyme? Is it coming from the water? Is it added before the reaction, after the reaction, during the reaction? The reason I don't care at all where that proton comes from is because so many different reaction mechanisms pull the proton from so many different places and at so many different times that that's not useful to memorize. In fact, Can you even imagine how much effort would be involved to memorize where that proton comes from? In every reaction we look at, it's much more powerful to say, wow, water's chock full of protons. I really don't care where it comes from. It's going to be easy to get the proton. What is powerful is to focus on just the general overall mechanism, which then can be broadly used and does not require so much brute force memorization. A fundamental thing about this reaction is its name, hydrolysis, says water. The water molecule actually becomes part of the product. Therefore, you begin with 2 oxygen atoms in an ester bond. The product, there are 3 oxygen atoms, one of them coming from the water, which means that in most organic reactions involving a nucleophilic attack step, you're either going to increase the number of oxygen atoms by 1 or decrease the number of oxygen atoms by 1. In the case of hydrolysis of an ester bond, those 2 products will specifically have a carboxylic acid group and an alcohol group, basically a hydroxyl. In the specific case of hydrolysis of an acetyl group from a compound, that carboxylic acid will be specifically acetic acid. Acetic acid is profoundly important because it reminds you that the acetyl group has 2 carbon atoms. So acetic acid has 2 carbon atoms and 2 oxygen atoms, and you really need to know that because acetyl groups are constantly being donated as part of metabolism. This 2 carbon atom group is donated in the case of fatty acid biosynthesis, cholesterol biosynthesis, ketone body biosynthesis, how it is taken away in the case of fatty acid oxidation or ketone body oxidation to make energy. This 2 carbon acetyl group is donated to DNA in order to regulate gene expression. Acetyl groups are donated to sugars in order to build various different types of oligosaccharide signaling molecules for the immune system. Therefore, to help you remember that acetic acid is a very simple acid with 2 carbon atoms and 2 oxygen atoms, I want to remind you that acetic acid is vinegar. Can you sort out the etymology of that word? Vine, gar. Ever hear of Vinland, Vineland, the Vikings and all? Vine means wine, of course. Gar actually means sour. Vine, gar, sour. Acet, acetic, sour acid. Vinegar is what makes wine sour. So, when you open a bottle of wine and let it sit for a day or two, where does the vinegar come from? Please pause and answer. Well, I'll give you a clue if you need it. There is a compound that's prevalent in wine and contains 2 carbon atoms to begin with. And in fact, this compound is deeply involved in liver damage and fatty liver disease. How about ethanol? Ethanol, ethane, 2 carbon atoms, all hydroxyl group, can be oxidized to acetic acid. When you open a bottle of wine, oxygen in the air gets in there, and microorganisms can use that oxygen to oxidize ethanol to acetic acid. In fact, these same oxidation reactions are catalyzed by the human liver in order to process alcohol when you drink it. Will you please invent a name for this enzyme? To be clear, I'm just hoping that you make up a name that fits the rules of enzyme nomenclature. That doesn't mean that the name you come up will exactly match the technical word that is currently used. The idea is, can you interpret the names, and then once you know the name, it'll be easier to remember it. One hint is it's going to be substrate followed by activity. So the substrate is acetylcholine. What is the activity? Well, we have an ester bond that is hydrolyzed. And the technical name is acetylcholine esterase. It breaks an ester bond. You're going to find out that acetylcholine esterase is an extremely important enzyme involved in nerve signaling. But as I've already said, the general mechanism which describes how acetylcholinesterase works is much more broadly applicable. So this exact same mechanism of hydrolysis is a way that peptide bonds can be broken when you digest protein in your diet or in a cell when it's degrading old proteins. Genomes can be broken down one nucleotide at a time by hydrolysis. Ester bonds can be broken, thiol esters, amides. Phosphate groups can be hydrolyzed off of alcohols. In addition, just as you can hydrolyze all these bonds, many reactions begin with a nucleophilic attack step, such as the assembly of amino acids into protein, the assembly of monomeric nucleotides to make RNA molecules, the assembly of deoxynucleotides to make genomes, and as well as others. So this is a very powerful reaction to be familiar with. In fact, this mechanism is so important, that if I ever give a talk where I fail to use a nucleophilic attack mechanism to describe the underlying chemistry, whoever catches me first will get a prize. If you want to ensure that you get the most benefit by my description of these nucleophilic attack mechanisms, which will be used in every lecture, I'd like you to look at some of the nomenclature of the reaction. For example, please identify the nucleophile and the electrophile in the substrates for hydrolysis of acetylcholine. Go ahead and do that before moving on. And again, through all of my talks, you will get a lot more out of them if you actually attempt to answer the questions before advancing. If you simply advance through the whole talk, I really think you're pretty much wasting your time. And again, I'm always open for questions. A nucleophile is a lover of the nucleus. An electrophile is a lover of electrons. Of course, electrons are negatively charged, so the lone pair of electron will repel negative charges, but will be attracted to the nucleus. So the nucleophile, the lover of the nucleus, the lover of the positive charge, are the negatively charged electrons. Meanwhile, the electrophile, the lover of the electrons, is the partially positively charged carbonyl carbon. So the nucleus is partially positively charged because the nucleus is positive in the first place, but furthermore, this nucleus is linked to two very electronegative oxygen atoms, both of which will inductively pull electrons away from the central carbon, making it more positively charged and a better electrophile. Here I'm showing the overall reaction mechanism where the nucleophilic attack has caused the electrons to resonate up onto the oxygen to form an intermediate. We know that carbon always can only have 4 bonds to it. So if the electrophile is going to form a bond to the carbonyl carbon, then the electrons in the double bond to the oxygen have to resonate up on there. Now, between the substrates, intermediate, and products, can you identify the oxyanion? Please pause and do that. And if you're a little bit confused, just interpret the name. Okay? I'm trying to make a point that everything is what it's called. The oxyanion is an oxygen atom anion with a negative charge. It's real obvious what the oxyanion is. In addition, you can see how the nucleophilic attack takes place. Can you now show the movement of electrons in order to convert the intermediate into the product? Please draw the arrows. And in every one of these nucleophilic attack steps, there's going to be 2 arrows showing the attack and 2 arrows showing the resolution. Please do so. Well, what happens is, as the electrons resonate from the oxyanion to reform the double bond, that will force the electrons linking the central carbon to the choline to resonate up onto the oxygen of the choline, which will then pick up a proton. From where? I don't know and I don't care, but it'll grab a proton and you now have the choline and you now have the acetic acid. And when I see this, I ask a question. Wait a minute. When the electrons resonate down, why does it kick off the choline? Why not just kick the water molecule back off? And the answer to that is, there's no reason. In fact, this intermediate can resolve in either direction equally. But the point is, if you have a bunch of acetylcholine and you have the enzyme forming the intermediate, about half the time it will reform acetylcholine, and about half the time it will form the choline and acetic acid, which means the net effect is to tend to convert acetylcholine to choline and acetic acid. Now, to be clear, breaking something apart goes down in energy. So the favorable reaction will be toward dissociation. But from the intermediate itself, the reaction can go in either direction. But if you have a whole bunch of substrates, you're going to tend to form mostly products. I'd like to be clear on 2 points now. I've really delved into this one reaction in detail, and I wanted to be sure that whatever your level of organic was, you'd probably understand it. Right now, I'm going to give you my cell phone number, and please write it down, 843-225-5022. I'm giving you this number because I sincerely hope you use it if you get stuck on some of this. If you've been working on this and something doesn't quite add up or you're confused about some of the terminology, I would like to have the opportunity to help out. I'm really hoping you don't get bogged down in this, and I would like to help you through it more quickly. The more confused you are and the less certain you are about what to ask, the more important it would be to talk with me and sort it out quickly so that then you can advance and move on to higher levels. So don't hesitate to call. Now that we've gone into such detail on the nucleophilic attack, I'm going to finish the talk by looking at something that's less important, but still matters, and that is the fact that this carboxylic acid that was released from the ester bond still has a proton on it. And in fact, I would look at an ester bond and a carboxylic acid as almost the same thing. An ester bond links two carbon-containing molecules together. When you hydrolyze it, you still do have one of those carbon-containing molecules, but instead of the other side of the link, you simply have a proton. Now, a carbon to oxygen bond isn't going to randomly break, but it is very easy for a proton to come off a carboxylic acid to form acetate. And in fact, part of the nomenclature, you really want to know that a conjugate acid, an acid is a proton donor by definition, so it has a proton, where a conjugate base tends to end with the word ate, meaning a negatively charged carboxylic acid that has lost its proton. And in fact, if you have acetic acid and acetate in your body, will there be more of the acid form or more of the base form? Please answer and explain why. Carboxylic acids, importantly, tend to lose their proton and exist primarily in the conjugate base form because losing the proton allows resonance and spreading out of the negative charge. If you've understood this talk, you now understand why hydrolysis of an ester bond tends to give products, one of which is an alcohol and one of which is a carboxylic acid. I'm going to now suggest you do a little exercise where you hydrolyze an amide bond through a nucleophilic attack mechanism and where you hydrolyze a thiol ester from acetyl coenzyme A using a nucleophilic attack, and tell me what the products are. I appreciate your time. Good luck with all of this, and I look forward to seeing you in class shortly. Thank you.